The 1980s marked the rise of nanotechnology, a field focused on manipulating matter at the nanoscale to develop materials and systems with unique properties. From the early 2000s onward, NASA began integrating nanotechnology research into its missions, recognizing its potential to enhance spacecraft subsystems. These fall into four main categories: spacecraft materials and structures, nanosensors and instrumentation, micro and nanorobotics, and life support systems. How, or rather why, can we use nanomaterials in such applications? For spacecraft materials and protective gear, nanomaterials such as carbon nanotubes, graphene, and nanocomposites have a high strength-to-weight ratio that provides mechanical strength while keeping structures lightweight, reducing launch costs. These materials provide enhanced radiation resistance, thermal stability, impact protection, and flexibility, making them ideal for spacecraft exteriors, habitats, and astronaut protective gear. Boron nitride nanotubes, for instance, are actively researched by the Space Agency's Langley Research Center and the National Institute of Aerospace for future use in protective systems, including radiation-resistant shielding, spacecraft hulls, crew habitats, and high-performance spacesuits. Meanwhile, a carbon nanotube composite overwrap pressure vessel has already been deployed by the Space Agency, reinforcing the outer layers of a pressure vessel with carbon nanotubes and creating a stronger, lighter vessel. As for nanosensors and instrumentation, nanosensors provide data and feedback by monitoring spacecraft cabin air and life support systems, detecting fuel leaks on launch pads, and analyzing planetary soil composition at the nanoscale. This is because nanomaterials conduct heat and electricity efficiently at very small scales, and their higher surface area, low energy barriers, and excellent thermal and mechanical stability allow nanostructured chemical sensors to provide greater sensitivity, reduced power requirements, and enhanced durability, making nanosensors, filters, and catalysts more efficient than macroscale equivalents. As early as 2007, the Space Agency developed a nanosensor chip using carbon nanotubes and nanowires, coated or doped with polymers and catalytic metals, which was successfully tested in the space to detect various gases. When a target chemical interacts with these nanostructures, it alters the electrical current, generating a distinctive pattern that allows the sensor to identify the specific chemical. With regard to micro or nanorobotics, robotic systems can be manufactured at the nanoscale and adapted for space exploration. Their motion is generated via chemical reactions, biohybrid propulsion, or magnetic light, ultrasound, or electric fields. Although there are no fully operational nanorobots performing autonomous tasks in space, current research seeks to integrate nanorobots for on-demand patching or maintenance of Martian spacesuits. One of the most directly applicable areas for terrestrial use, however, is the adaptation of nanotechnology-based life support systems for space. These systems include water purification, air filtration, and food preservation. Such mechanisms cannot be directly mapped into extraterrestrial applications, which sets the groundwork for regenerative, self-sufficient, and low-pressure systems.
Within this broad scope of life support strategies, the second pillar, water purification, emerges as the most viable pathway for extraterrestrial deployment. Water purification in space is both more critical and more challenging than on Earth. Frequent water supply to space stations is impractical, costly, and logistically challenging. Therefore, astronauts depend on closed-loop regenerative systems, where a limited water supply is continuously recycled and purified from sources such as urine, sweat, hygiene wastewater, and condensation, all of which contain salts, microbes, and chemical contaminants that pose health risks. In microgravity, contaminants cannot be separated naturally by gravity, making specialized purification technologies essential. These systems integrate distillation, evaporation, filtration, catalytic oxidation, and ion exchange to maintain a safe and sustainable water supply. For example, in the urine processor assembly on the International Space Station, urine is treated through vacuum distillation, where the low-pressure environment causes water to evaporate, condense, and be collected separately. Since there is no gravity or normal pressure differences, mechanical pumps are required to move water through filters and keep the system operating. However, pumps are heavy, energy-demanding, prone to failure, and generate heat and vibrations, making them difficult to maintain. These challenges highlight the potential of nanomaterial-based systems. Why nanomaterials? Their high surface area to volume ratio increases chemical interactions with contaminants, enhancing adsorption and reaction efficiency. Nanomaterials can also improve membrane selectivity and flux, permitting the passage of water while excluding impurities. Their catalytic properties also enable the degradation of organic pollutants, and their tendency for self-assembly can simplify fabrication processes, reducing manufacturing costs. While numerous technologies can leverage these properties for water purification in space, the key ones include three: nanoabsorbents, nanofiltration membranes, and acoustic nanotube technology. Although none are fully operational in space yet, nanofiltration membranes are the closest to potential application. Nanofiltration membranes are a type of pressure-driven filtration technology with pore sizes between 1 and 10 nanometers. Their selectivity surpasses that of ultrafiltration, removing salts, organic compounds, and larger molecules, but still less restrictive than reverse osmosis, since monovalent ions can still permeate. Nanofibers with diameters ranging from 10 to 500 nanometers, fabricated from polymers or composite materials, form the highly porous interconnected network that function as the filtration membranes. These mainly include electrospun polymer nanofibers among carbon nanotubes or silver and titanium dioxide. Pores in the nanofiber network are small enough to block larger molecules like dyes, proteins, or bacteria, while their hydrophilic surfaces and functional groups can repel ions or charged organic molecules. On Earth, this is achieved by pumping contaminated water at high pressure through a semi-permeable membrane. It's dependent on hydraulic pressure overcoming osmotic pressure to move clean water through the membrane. This is ineffective in microgravity environments where the lack of buoyancy and hydrostatic pressure inhibits conventional water movement and contaminant separation. In contrast, nanofiltration systems designed for space exploit surface tension-driven flow. At the nanometer scale of the membrane pores, water molecules are strongly attached to the hydrophilic interior, generating capillary action. This effect, combined with electrostatic interactions and nanoconfinement, accelerate water transport, enabling efficient filtration without reliance on pumps or external pressure. A NASA-led initiative by Langley Research Center aims to develop a nanofiltration system for long-duration human space missions beyond low Earth orbit. The system utilizes surface tension-driven flow to provide a low-power, high-speed alternative to the energy-intensive and time-consuming distillation or reverse osmosis methods within the next 2 decades. Another long-standing water purification technique adapted for space is the use of nano-absorbent materials. Traditional water-purifying materials such as activated carbon, clays, and waste-driven sorbents remove heavy metals and toxins. However, limitations like low absorption capacity reduce their efficiency. Over the past decade, nanomaterials have been explored to overcome these shortcomings. While nanofiltration membranes act as pore-based sieves, separating contaminants by size and charge, nanoabsorbents function like chemical magnets, capturing contaminants on their surface. Functional coatings or surface groups enable selective binding of specific molecules, enhanced by nanomaterials' high porosity and tunable pore structure. Common nanoabsorbents include metal oxide nanoparticles, which degrade organic pollutants or inactive microbes, carbon-based nanomaterials, effective for removing heavy metals and dyes, and polymer-based nanostructures or composites, functionalized with reactive groups for high absorption capacity. Magnetic nanoparticles are especially useful as they allow easy regeneration and recycling through magnetic field. The synthesis method determines particle morphology and surface functionality, both of which influencing efficiency. The top-down approach breaks bulk material into nanoparticles via milling, lithography, or laser ablation, which can produce inconsistent absorption sites. In contrast, bottom-up synthesis, through chemical vapor deposition, sol gel processing, or biological routes such as plant extracts, enables precise control over particle structure and surface chemistry, yielding more uniform and efficient absorbance. However, nanoabsorbents must be tailored for space, where gravity-dependent purification systems don't work as they do on Earth. Magnetically recoverable nanomaterials are especially useful because they can be easily collected in microgravity and reused in closed-loop water systems. Some nanomaterials can even regenerate their absorption capacity with light or mild heat. Combining different nanoparticles such as photocatalytic and absorptive types also enables simultaneous capture and breakdown of contaminants. Once used, though, these nanoabsorbents can become hazards themselves if not properly treated or regenerated. Techniques used for non-toxicity include heating, steam, or chemical treatment, microwave energy, applying voltage to release ions, or microorganisms to naturally degrade pollutants, all of which are adjusted according to pH, temperature, and contact time to optimize recovery. In terms of water, the last of the three main technologies is acoustic nanotube technology. Developed by NASA for astronauts aboard the ISS, it employs sound waves to induce water flow rather than relying on pressure-driven filtration. Its core component is a molecular scale membrane of vertically aligned carbon nanotubes. These are hollow cylinder nanostructures with pores that permit water molecules while blocking dirt, bacteria, and chemicals. The surrounding matrix can be composed of ceramic, polymer, or metallic materials. How it works is an oscillator circuit attached to the filter generates vibrations that debond water, increasing mobility and pushing them through the pores without relying on gravity or pressure. The waves drive clean water molecules away from contaminants rather than trapping them, which reduces clogging and extends the filter's lifespan. Some particles may accumulate near the inlet, reducing water flow. When the output flow drops below a preset threshold, a cycle automatically activates, which removes sediment from the inlet, so no manual flushing, like conventional filters, is required. But why carbon nanotubes? These structures have an incredibly high strength-to-density ratio. Their fibers are the thinnest known, with diameters about 30,000 times smaller than a human hair. The development of single and multi-layer transparent carbon films in 1991 marked a pivotal step in harnessing the acoustic and filtration properties of CNTs for applications such as water purification and microgravity. Yet water is only one part of a closed-loop system. The same space environment that requires efficient water recycling also requires precise air control. But how does air filtration work with these systems?
Air in a spacecraft is a closed loop. Every breath adds carbon dioxide and water vapor. Materials, electronics, and experiments release trace volatile organics. People and equipment shed tiny particles and bioaerosols. The job of air revitalization is to remove all of that continuously with very little power and very little crew time. Nanobiotechnology helps because it lets us engineer the interfaces the air actually touches, such as fiber diameters, pore size, surface charge, and catalytic sites, so filters capture particles more efficiently. Catalysts will be able to destroy volatile organics rather than just store them, and sorbents keep holding carbon dioxide even when the air is humid. At present, the International Space Station follows a three-stage architecture. First, a fibrous particulate filter protects crew and hardware from dust and bioaerosols. Pressure drop is monitored because it drives fan power and elements are changed on schedule. Second, a trace contaminant unit combines activated charcoal with a heated catalytic oxidizer in order to remove formaldehyde, acetaldehyde, alcohols, and aromatics. Those beds must be replaced or regenerated. Third, a 4-bed molecular sieve assembly cycles continuously. One desiccant bed dries the airstream, while one carbon dioxide sorbent bed captures carbon dioxide. The offline pair is heated and vented to space to store capacity. This sequence is robust, but it costs power through pressure drop, loses capacity in humidity, and relies on expendable elements and crew labor. For particulates and bioaerosols, we propose electrospun nanofiber media. Submicrometer fibers create very small pores and very large surface area, so diffusion and interception dominate in the hardest size range, including ultrafine particles and viral scale aerosols, while overall pressure drop stays low. This results in the same as clean air delivery at lower fan power and longer intervals between changes. The mat can be reinforced with graphene oxide or carbon nanotubes for stiffness and mild conductivity, then immobilized in a bonded scaffold and verified for zero shedding with downstream particle counting over thousands of hours. For trace volatile compounds, the goal shifts from storage to destruction. A photocatalytic stage coats a duct panel or porous support with titanium dioxide nanoparticles. Under visible LED light, the surface generates reactive hydroxyl species that mineralize a broad spectrum of organics all the way to carbon dioxide and water. That reduces consumables because contaminants are converted in place, not loaded onto charcoal. In order for this to work, the catalyst is tuned for visible light. Permanently bonded to the substrate, the flow path is designed for uniform illumination and residence time, and sensors confirm complete conversion under humid, mixed contaminant streams. For carbon dioxide control, which is the largest sink of energy and mass, we augment or replace zeolites with humidity-tolerant metal organic frameworks. These crystalline ultraporous materials let us tune pore size, hydrophobicity, and binding chemistry so carbon dioxide capacity stays high even when water vapor is present, and regeneration can happen at lower temperatures or gentler pressure swings. A zeolitic imidazolate framework keeps almost the same breakthrough time in dry and very humid streams, showing that working capacity is preserved when the gas is wet. For flight, we press the material into strong pellets or a honeycomb-style monolith to cut pressure drop, add a small moisture buffer layer upstream, and target longer absorption windows with cooler regenerations. To prove these upgrades at a system level, we use operational metrics. For particles, clean air delivery at a given pressure drop should have the same or better cleanliness with less fan power and longer service intervals. For volatile organics, mineralization efficiency to carbon dioxide and water, byproduct speciation, and added pressure drop with continuous polishing with fewer expendables. For carbon dioxide, the breakthrough time, working capacity in humid air, and energy per regeneration, it should allow for longer absorption windows, shorter, cooler regenerations, and beds that remain mechanically intact over thousands of hours. Across all components, we enforce safety and durability, no nanoparticle release, stable performance under humidity and radiation, and predictable end-of-life behavior. In summary, today's loop keeps the cabin safe, but at the cost of power consumables and humidity sensitivity. Our nanobiotechnology approach, such as electrospun fiber media, photocatalytic destruction of trace gases, and humidity-tolerant metal organic frameworks, which target those exact limits, yielding an air revitalization system that is lighter, quieter, and more self-sustaining, and that sets the same design logic we will use for water recycling and food preservation.
Every year, nearly one-third of all the food produced globally is lost because it spoils. Traditional packaging like plastic, glass, and metal can protect food from the outside environment, but it cannot actively preserve it, and it cannot sense freshness. Nanobiotechnology completely transforms this. By working at the nanoscale, between 1 and 100 nanometers, it gives materials new abilities, the ability to fight bacteria, detect chemical changes, and protect food in natural and sustainable ways. What makes this field so unique is how it blends biology, chemistry, and material science to create packaging systems that behave almost like living organisms, responsive, adaptive, and intelligent. One of the strongest applications of nanobiotechnology in food preservation is active protection through antimicrobial nanoparticles. Materials like zinc oxide, silver, and titanium dioxide have powerful antimicrobial effects that prevent bacterial and fungal growth, which are the main causes of food spoilage. They work through several complementary mechanisms. Some generate reactive oxygen species that damage bacterial cell walls and DNA, others physically disrupt microbial membranes by attaching to their surfaces, and some release ions such as zinc or silver which interfere with essential cellular processes. When these nanoparticles are embedded into biodegradable films or coatings, the packaging becomes an active defense system. Instead of passively wrapping food, it becomes a microscopic shield that can kill bacteria, slow oxidation, and significantly extend shelf life. This brings us much closer to packaging that does more than protect. It interacts intelligently with the food it contains. But antimicrobial effects alone aren't enough. Sustainability is equally important. This is where natural nanostructured materials come in. Researchers use biopolymers such as zein, cellulose, and lignin, materials derived from plants, and structured them at the nanoscale to create biodegradable and eco-friendly packaging. Through a technique called electrospinning, they can form extremely thin and porous nanofibers. These fibers can hold antioxidants or essential oils that slowly release over time to delay oxidation. They act as effective barriers against oxygen and UV light, and after use, they decompose safely without harming the environment. These plant-based nanomaterials show that high performance and sustainability can go hand in hand. They redefine packaging as something functional, renewable, and compatible with circular food systems. Another major advancement is intelligent packaging powered by nanosensors. This is the transition from simply protecting food to actually monitoring it in real time. Nanomaterials like graphene, gold nanoparticles, and carbon nanotubes can detect tiny amounts of gases such as ethylene or ammonia, which are natural indicators of spoilage or ripening. Other nanosensors react to changes in pH, humidity, or volatile organic compounds linked to microbial activity. Some can even detect specific pathogens or toxins using optical or electrical signals. This allows packaging to communicate freshness visually. Imagine a milk container that turns blue when bacterial levels rise, or a fruit label that changes color as ripening progresses. This approach helps consumers make better decisions, reduce unnecessary food waste, and increases food safety, all without additional technology. Currently, in space missions, food preservation relies on thermal sterilization, freeze-drying, vacuum sealing, and limited refrigeration. Most space foods are dehydrated or thermostabilized and stored in multi-layer polymer pouches. These methods are effective for short missions, but they also create challenges. High temperature processing can reduce nutrients and flavor. The packaging is bulky and non-biodegradable, which produces waste. There is no real-time spoilage monitoring, meaning astronauts cannot easily confirm if a sealed package is still safe, and refrigeration is extremely energy-intensive, making it impractical for long missions or deep space travel. This is where nanobiotechnology becomes a game changer. Antimicrobial nanocoating could reduce or even eliminate the need for extreme heat sterilization. Smart nanosensor-based packaging could automatically detect spoilage and signal safety through visible color changes. Lightweight, plant-based nanostructured films could replace heavy plastics, reducing both launch mass and waste. Because these materials are biodegradable and responsive, they integrate naturally into closed-loop life support systems in spacecraft and future lunar or Martian habitats. In future space exploration, where every gram of waste matters and resupply is limited, nanobiotechnology could fundamentally reshape how astronauts store and monitor their food. It could extend shelf life, lower energy consumption, and provide instant indicators of freshness, something especially important in microgravity, where smell and visual cues are unreliable. Ultimately, nanobiotechnology unites two worlds. It helps us preserve food more effectively and sustainably here on Earth, while also building the foundation for reliable, self-sustaining food systems beyond our planet. It isn't just about keeping food fresh, it's about redefining preservation as an intelligent, dynamic process that supports human life wherever it exists.
Although the most well-documented life support strategies for space remain in the proposal or experimental phase, numerous nanotechnological tools and techniques are already in active use. These include atomic force microscopy for material characterization, electron beam lithography for fabricating nanoscale patterns in sensors and electronics, as well as nanofabrication methods and simulation softwares designed to predict the behavior of nanomaterials under space conditions. The ongoing use of nanocomposites in spacecraft materials or as coatings for radiation shielding and nanosensors in satellites and rovers highlights the potential for developing multifunctional, nanocomposite, and intelligent adaptive systems. Artificial intelligence in particular is gaining traction in the field. By leveraging complex algorithms with nanotechnology's precision, we can further the adaptability of such systems in microgravity environments. Although the proposed methods already bypass the primary challenges of decontamination and water flow in space, fluid behavior, microbial growth, and system degradation can still be unpredictable. Real-time monitoring, adaptive control, and predictive maintenance with smart nanosensors would allow adjusting system parameters, automatically adjusting flow rates, adjusting photocatalytic reactions, or monitoring saturation levels of nanoabsorbents. Additionally, AI can optimize the allocation of critical resources, power, oxygen, and water, addressing the constraints imposed by limited supplies in space missions. On Earth, these advances could translate to compact and energy-efficient water treatment plants, smart air purification systems, or shelf-stable food packaging with minimal energy input.
To transform these nanoscale technologies from laboratory research into real-life support systems for space, several key conditions must be met. This requires close collaboration between nanomaterial scientists, microgravity experts, robotic engineers, and space mission planners. Safety must be managed through dedicated nanomaterial risk systems, while scalability, cost control, sustainability, and long-term resource usage must all be optimized. Prototypes must also be tested repeatedly under simulated space conditions. Most importantly, solutions proven on Earth cannot simply be transferred to space. Although they provide valuable guidance, this field must be treated as a distinct scientific domain with unique environmental challenges and exciting new opportunities.